Alright folks, today what we're going to look at real quick first is go back through active transport and make sure that we understand what that is, okay? Now we've already talked about a little bit about active transport. Remember we said that active transport requires us to have energy in order to do what we've got to do. So we have to have some ATP to get the cell to do what it needs to do as far as moving stuff in and out, okay? And what we usually do is when we move stuff from low concentration to high concentration. What that basically means, instead of going down the gradient, where we go from high to low, we're going up the gradient. We're going from low to high. So we're basically pumping stuff in or pumping stuff out, and that's requiring us to use energy to get that. So we're going from, well, we got a little bit right here, and we got a lot out there. Let's take this little bit and pump it out to where all the other stuff is at, where there's a lot of stuff. And again, that usually goes against, that's going to go against the gradient. And one of those that we mentioned was something called the sodium-potassium pump. And this pump, again, as I mentioned yesterday, was used a lot, or is used a lot by our nerves to help us to move around and do things. And without this particular pump of pumping sodium and potassium in and out of the body, our ability to do things would be non-existent. We wouldn't be able to do stuff. We wouldn't be able to move and things like that. So this is extremely important in our everyday lives because we're using it every second of every day. Okay? And as I said and showed you, we pump out sodium, we pump in potassium. Okay. Now let's talk about moving some bigger things in and out of the cell besides just ions. Okay. Let's talk about moving the big stuff out of a cell. Say we want to get rid of some stuff that we don't want, some waste products. We take it, we put it in a vacuole, this vesicle, basically an empty bubble, fill it with our stuff we don't want, or the stuff we got to get out, and we shove it up towards the edge of the cell membrane. Then it joins with the cell membrane, and all that stuff, in this particular case, proteins, goes out of the cell. This is called exocytosis. And again, exo meaning exiting, cyto meaning cell. We're exiting the process of exiting the cell. So molecules are moved out of the cell by vesicles that fuse or join with the cell membrane. So if we want to get big stuff out of the cell, we basically push it out through these little vesicles, and then it goes out of the cell and goes to wherever it's got to go next. Okay? So that's exocytosis. We're exiting the cell, right, with the materials that we want to exit. Now, what we have in there is exocytosis. See there, it goes out. This vesicle comes in, joins with the cell membrane. All those proteins or whatever go out, and this becomes part of the cell membrane again. Okay? So this is happening every second of every day, hundreds of thousands of times a second in those cells, moving stuff out of the cell. Now, this occurs very rapidly, and it occurs quite often every day. Now, inside the cell, outside the cell, right? This is exocytosis here. The cell membrane is literally, this is the cell, the vesicle, pumping this material out into the environment where it needs to be. Okay? Now, the exact opposite is endocytosis because we're bringing stuff into the cell. So here you can see step one, look what's happening. Here's the cell membrane, here's some stuff. And look at how it starts to fold in around that stuff and then forms a bubble. And that bubble, that vesicle, pooches in, and now we've got a vesicle on the inside of the cell that's got stuff that we want. Okay? So this is literally the opposite of exocytosis, endocytosis. Okay? Endo meaning inside, cyto cell, osis the process. So going in, the process of going inside the cell. Okay? So this happens on a day-to-day basis, every second of every day, thousands of times there in the cell. Okay? Now there are two types of endocytosis. It depends upon what we're pulling into this cell. Okay? If we're pulling in liquids, water materials, like this is called pinocytosis. Pinocytosis, we're pulling in liquids into the cell. Okay? We a lot of times call this cell drinking because we're literally drinking liquids and pulling them into the cell. Like you and I drinking a Coca-Cola, right? Or soda of some sort, or tea, whatever. We're bringing it in liquid form, and there's nutrients in the liquid, right? This is what's happening in pinocytosis. Cell drinking. Easy to remember, right? right? So again, you can see a picture here. They've actually highlighted this so you can actually see the cell membrane and then these little vesicles forming with the liquid inside. This is pinocytosis. These will go inside the cell and be used by the cell for whatever the liquid is that's being pulled in, right? And again, the opposite of pinocytosis, instead of bringing in liquids, we bring in solids. This is called phagocytosis. Okay? So think of this bacteria right here. This bacteria is getting surrounded and eventually pinches up into a vesicle and goes inside the cell. This is called phagocytosis. Large particles, food, bacteria are engulfed. They're surrounded and eaten, if you want to call it. That's what we call it, cell eating. They're eaten by the cell. Then the cell will take those, remember those lysosomes? It'll take those lysosomes and break them apart into smaller pieces and then divvy them out to the cell so the cell can use those pieces of material. Okay? So we have exocytosis, endocytosis. Exo meaning out of the cell, leaving the cell, endo bringing in. We can breathe in liquids, pinocytosis, or we can bring in solids in phagocytosis. So again, this is not going to work. There it goes. It swallows it. There it is. It's coming in, and there comes old lysosome come along, and then dissolves it and eats it up. And again, this happens all the time in your body, hundreds if not thousands of times per cell on a daily basis. And this is all that's going on. That bacteria or that piece of food is being dissolved. So that's phagocytosis. Okay? And here's a cool, this is a picture under an electron microscope of a cell engulfing. See, this is kind of a weird looking little critter, isn't it? But this single cell critter is actually shooting a piece of itself forward, and it's about to surround this bacteria and eat it. It kind of looks like some weird creature basically slurping itself forward, wrapping around the bacteria, and then dissolving it, pulling it in and dissolving it. But that's basically what it's doing. But I always thought that picture was kind of cool looking because it looks like some sort of weird, monstrous creature, some blobby creature coming along. It even looks like it's got eyeballs over here and a big giant proboscis nose, right? Now, that's active transport. There'll also need to be another little video on active transport on there as well, okay? So you need to make sure that you're looking for these later today, okay? Part 2 will be on here as well. We'll talk about the different kinds of solutions, okay? All right.